All right, everyone. For a long, long time, we've thought of the genome as the ultimate biological blueprint, the book of life. But what if that script is only half the story? In this explainer, we're going to jump way beyond just the DNA sequence. We're diving into the omics revolution. These are the technologies that let us actually watch the biological performance as it's happening and really start to understand what makes a cell tick. So here's a quick look at what we'll cover. First, we'll talk about why the genome sequence on its own just isn't enough. Then we'll follow that cascade of information from DNA all the way to function. After that, we'll get into the weeds and look at the huge technical challenges these omics technologies throw at us. And finally, we'll see the big payoff, how we can use this to map out the incredible molecular machinery inside a cell. Okay, let's get started. When the genomic era first kicked off, there was this incredible sense of promise. The thinking was that if we could just read an organism's entire DNA sequence, we'd basically unlock all of its secrets and solve some of the biggest problems in biology. And this quote from Barry Bloom back in '95, it just perfectly captures that feeling, that sheer optimism. The idea was nothing short of revolutionary. Just sequence a pathogen and bam, you'd have a complete parts list. Every virulence factor, every single antigen, every possible drug target. It was this idea that we could literally engineer our way out of diseases just by having the sequence. But pretty quickly, a really critical question started to surface. How much does a static list of A's, T's, C's, and G's really tell you about a living, breathing, dynamic organism? Because think about it, the things we actually want to understand and stop, like toxins or enzymes or those drug resistance pumps, they aren't DNA. They're the products of those genes. And that's really the heart of the problem. If the genome is the script for a play, the actual performance, what we call the phenotype, is carried out by the actors, the proteins and the metabolites. Now, the script is absolutely essential, don't get me wrong, but it doesn't tell you how the actors are going to deliver their lines, or what costumes they'll wear, or how they're going to interact on stage in real time. Our challenge is to figure out how to watch that live performance. And that right there is exactly where omics technologies come in. They're the set of tools that let us finally move past just reading the script and actually watch that performance, layer by molecular layer. So let's break it down. At the very bottom, we've got the genome. This is the master blueprint, right? The complete set of genetic instructions. For any given organism, it's pretty static. It's stable. It holds all the potential information. Now, the very first step in bringing that blueprint to life is creating the transcriptome. This is the collection of all the RNA molecules in a cell, and it tells you which genes are actively being read from the DNA at one specific moment. You can think of it like a real-time memo, showing which instructions are being sent out for use right now. And from those RNA instructions, we finally get to the proteome. This is the entire constellation of proteins that are actually doing stuff in the cell. These are the real workhorses, the enzymes, the transporters, the structural bits. They're the actors executing the script, and they're what truly define what a cell is doing and what it's capable of. This diagram here just lays it all out so well. You see at the top, we've got the static genome that gets transcribed into the transcriptome, which is way more dynamic, right? It changes depending on what the cell is up to. That then gets translated into the even more complex proteome. And then finally, those proteins act as enzymes, making and changing tons of small molecules, which gives us the super dynamic metabolome at the bottom. The key takeaway here is this shift from a single static blueprint to these incredibly responsive, constantly changing layers that actually reflect the cell's real life. So the goal is to measure all of these different layers. But it turns out that as we move down this cascade from DNA to RNA to proteins, the chemistry of the molecules themselves creates a bigger and bigger set of technical challenges. It's definitely not as simple as just reading a sequence. This table really spells out why it gets so much harder. Look at the genome. DNA is chemically uniform and super stable. Easy enough. Then we get to the transcriptome. RNA is a bit less stable, but, and this is the critical part, we can amplify it. We have this magic tool called PCR that lets us make millions of copies so we can find even the rarest transcripts. But when we get to the proteome and metabolome, whoa, the game completely changes. There's no PCR, and we're dealing with just this massive chemical diversity and a huge range in how much of each thing there is. It's a whole different level of difficulty. Let's just focus in on this protein problem for a second. First, like I said, there's no PCR for proteins. We can't just amplify a tiny signal. This creates what we call a dynamic range problem. It's like trying to hear a single person whispering in the middle of a packed football stadium. The superabundant proteins just drown everything else out. On top of that, there's the insane chemical diversity. It's not just 20 amino acids. Proteins get all these little modifications like phosphorylation or glycosylation tacked on after they're made. These act like molecular on-off switches, completely changing what the protein does. It's like trying to catalog every single vehicle in London, from a rusty bicycle to an A380, all at the same time. So you might be thinking, okay, if it's that hard, why on earth do we even bother? Well, the answer is that the scientific payoff is just enormous. Because when we tackle the proteome, we stop looking at individual parts and we start seeing how the cell's machinery is actually built, how it all works together as this coordinated, interacting network. And this is the field we call interactomics. See, proteins are social creatures. They rarely act alone. They get together to form these complex machines. The image you're seeing here, for instance, shows all the protein parts of a ribosomal subunit, all working together perfectly. These interactions drive pretty much every process in the cell. And here's the kicker. You simply cannot predict this complex web of partnerships from the gene sequence alone. No way. Proteomics gives us the tools to map these interactions directly, to finally see how these incredible molecular machines were actually built and controlled. So how do we actually do it? How do we find out who's friends with whom inside a cell? Well, a classic technique is called co-immunoprecipitation. Imagine you want to know what your favorite protein, we'll call it protein X, is hanging out with. You basically go fishing. You use a super specific antibody as your molecular hook to fish protein X, the bait, out of a big, messy soup of all the cell's proteins. The hook grabs the bait, and anything that's physically stuck to it comes along for the ride. Then you carefully wash away all the junk that's not specifically bound. Finally, you identify both your bait and its captured partners, the prey, using a technique called mass spectrometry. Think of it as a hypersensitive scale for weighing molecules to figure out exactly what they are. It's an incredibly powerful way to start building out those crucial interaction maps. And this brings us to the big final question. We started with a simple promise of genomics, and we saw its limits. But now we're armed with these powerful, if really difficult, omics technologies that let us see a cell's performance in real time. So how do we apply this? For us as microbiologists in our constant fight against antibiotic resistance, how can we leverage all this, proteomics, metabolomics, to find and validate the brand new drug targets that we so desperately need? That right there, that is the challenge and the huge opportunity that's waiting for us.